Good morning, everybody, and welcome to this special grand round. It is my pleasure to announce this year's J.P. Maclean Memorial Lecture that will be given by Dr. Janet Rossant, who is the 2016 International Friesen Prizewinner. We have a big program, so I immediately call on Dr. Obi Angel, who many of you will still know. He is one of my predecessors. And he will introduce the lecture and our lecturer. Obi, please.
Thank you very much, Dr. Renner. It's a great privilege to be here. Good morning, ladies and gentlemen. It's really a treat for me to welcome you as well to this very special Grand Rounds, which is a program devoted to the J.P. Maclean Memorial Lecture, now honoring Dr. Janet Rossant, the 2016 Henry G. Friesen International Prize winner. This day brings great memories to me of a personal significance, and I'd like to share this with you for a second. 26 years ago, almost to the day, in 1991, I chaired my first Grand Rounds here as the new head of internal medicine. It's déjà vu in a way, so it's kind of interesting to see the way progress is made. And congratulations to you, Dr. Renner, for taking the medal. And I wish you well in your new undertaking. Some 6 months after I began, Dr. John P. Maclean died, July 1991, of a massive heart attack at the young age of 65. This was a tragic loss to his family, to his patients, and to our department. I knew him when I was a medical student in 1957, when he taught endocrinology in the Department of Physiology under the leadership of Manitoba's iconic physiologist, Dr. Joe Dope. Dr. Maclean was a very popular and sensitive clinician, a devoted teacher, and an exemplary role model for medical students and residents. And in recognition of Dr. Maclean's humanism and academic contributions, the Department of Internal Medicine established this lectureship to honor his legacy. Dr. Maclean is also remembered for his pioneering interests in developing awareness of ethical issues in clinical medicine and was the first chair of the University Medical Ethics Committee. Biomedical ethics as a discipline has grown exponentially in the past quarter century and is now relevant in all areas of clinical science and clinical care now. Today, we hear about its impact at the frontiers of research in developmental biology, stem cell science, and regenerative medicine. This is an opportunity also to acknowledge the Maclean family, and Ian Maclean is here. Great to see you. Ian, thanks so much for being here. It's also a chance to acknowledge senior colleagues at the University of Manitoba who have been staunch supporters of Friends of CIHR and the Friesen Prize program. Henry Friesen himself is here. Thank you, Henry, for being a president, and congratulations to you. Of course, the award is in his name. Grant Pierce is here. Thanks for being here. Grant's a member of our board of directors. Fred Aoki, Narinjan Dalla, Arnold Neymark, and Alan Ronald. These are the people, charter members of our organization. Parenthetically, Fred Aoki was a member of the executive committee of the Department of Internal Medicine who in 1991 established the Maclean Lectureship. Nice to see you here, Fred. Now, a little bit about Friends of CHR, because many people don't know about what we do and how we spend our energy, but the Friends of CIHR is a national charitable organization and was established here in the Department of Internal Medicine in the year 2000. And its purpose is to advance the goals and ideals of CIHR and to establish partnerships in the community, and in so doing promote a better understanding of the importance of health research in society. Friends of CIHR has additional aims, which are to raise the level of discourse on health issues broadly and to encourage and support young trainees in their development as future health scientists. And a feature of the Friesen Prize program is to encourage the interaction of senior scientists with junior colleagues and early career scientists, just to promote their lives and well-being. Now, Friends of CIHR achieves their goals through three major programs. If I can advance this 2 slides, or 3 slides, do this manually somewhere. Next, there. We achieve our goals through 3 major initiatives: partnerships with health research societies, including the Canadian Academy of Health Sciences, Canadian Society for Clinical Investigation, Diabetes Research and Treatment Center, Banting Research Foundation, CBC, and others. We also have the Henry G. Friesen International Prize in Health Research, as you can see. As well, we have the Video History of Medicine in Canada project with Professor Michael Bliss. The Henry G. Friesen International Prize in Health Research has become our signature program and was established by Friends of CIHR in 2006 to enshrine Dr. Friesen's contributions in Canadian health research and health policy development. Dr. Friesen is famous for the discovery of the hormone prolactin and was instrumental in the creation of Canadian Institutes of Health Research. Over the years, the prize has expanded into a national program involving a number of Canadian universities and institutes. Now, you should know that in accepting a Friesen Prize, the laureate, Janet Rossant is the current, assumes a number of responsibilities. The prize winner undertakes a public lecture in Ottawa in conjunction with the annual meeting of the Canadian Academy. That person also prepares a manuscript for publication in the Friesen Prize lecture series. And the prize winner is also introduced by Paul Kennedy, the CBC Radio 1 Ideas for Future Broadcast. And I think next week, the Friesen Prize this year will be broadcast in that program. Finally, the prize winner undertakes institutional visits at major universities and research institutes across the country and shares their wisdom and experience with a wide audience of students, faculty members, and policymakers. This explains Dr. Janet's visit here. Of course, none of this can happen without the support and sponsorship of major institutional leaders in Canada, and I'd particularly like to acknowledge Dean Brian Postal, Dr. Eberhard Renner, and his predecessors for the generous support of the Friesen Prize program. The Faculty of Medicine here has been a staunch major sponsor since its inception. Now, it's my honor to introduce the special guest, Dr. Janet Rossant, who's the 2016 Henry G. Friesen International Prize winner. She's president and scientific director of the Gardner Foundation. She's a renowned developmental biologist. She demonstrated the origin of cells in the early embryo that can give rise to all tissues and the entire body of an intact animal, and she'll be telling us about this. Dr. Rossant's leadership skills were evident as Chief of Research of the Research Institute, the Hospital for Sick Children, from 2005 to 2015. She there spearheaded an integrated vision of scientific discovery, clinical application, and population impact. She also served as its scientific director of the Ontario Institute of Regenerative Medicine. In May 2016, she became the president of the Gardner Foundation. She was born in Chatham, Kent, UK, in the year, obtained her BA at Oxford University, PhD at Cambridge University, and in 1985, she came to the University of Toronto, first at the Lunenfeld Tannenbaum Research Institute at Mount Sinai Hospital, and from 2005 at the Hospital for Sick Children. She's published over 384 peer-reviewed articles and continues to publish, and has played a leading role in establishing guidelines for human embryonic stem cell research in Canada and beyond. Her many distinctions include fellowships in the Royal Society of London and Canada. She's a companion of the Order of Canada, recipient of the Michael Smith Prize, the Killam Prize, and the Gardner Whiteman Award. The title of Janet Rossant's lecture is Stem Cell and Genome Editing, Ethical Challenges in Human Health. Welcome and come to the podium.
Thank you, Orby, and it's an honor indeed to present to you today as the Friesen Prizewinner and also as the John Maclean Lecturer. I do not know Dr. Maclean, but he clearly is one of the many amazing clinical leaders that the University of Manitoba has had over the years, including my friend Orby Angel. So it's always a pleasure to be here at the University of Manitoba. It's a particular pleasure to give the Friesen Lecture, named after and in honor of Henry Friesen. And of course, Henry has made enormous contributions to research in Manitoba, so you can be extraordinarily proud of his continued commitment to the University of Manitoba, but of course beyond that in Canada more broadly. So his leadership of the MRC, and particularly his transition of the MRC to the AHRA, is something that has changed the face of health research in Canada. So Henry, congratulations on all you have done over the many years. So it's a great honor to be here. So what am I going to talk about to you today? So I'm going to give you a lecture that's basically very similar to the lecture that I gave in Ottawa. And it gives you a little bit of a taste of my research career. I'm going to give you my research career in about 3 slides. And then I'm going to tell you why my research has got me involved in ethical issues around human reproductive biology and human reproductive technologies, and where I see the Canadian situation around reproductive technologies and the impact of where science is going today. So, the title of the talk is Stem Cells and Genome Editing, Ethical Challenges in Human Health. But let's start at the beginning, and let's start with my favorite stage of development. This is the blastocyst. So this is what I work on. This is a mouse blastocyst. It's about 4 days of development. It's the size of a speck of dust. It's about 100 cells. If this was a human embryo, it would take 7 days to get to this point, to have a few more cells, but it wouldn't look very different. It wouldn't be as beautiful. Actually, the mouse blastocyst is much more beautiful than human blastocysts, but they both turn into whole organisms somehow along the way. That's the challenge for developmental biology, is understanding how you can go from a fertilized egg through stages like this, where there are clearly only a few different cell types, and no organization. This does not look like a mouse or a human being, but yet somehow the genetic information that's encoded in the genome is played out over time and allows these cells to differentiate and organize into a functioning organism. So working at this stage of development in the mouse, this is a stage at which we can culture the embryos from the 1-cell stage right through to this stage in a fully defined culture medium. So they think the embryo is self-organizing, it doesn't require any external signals, and yet it is beginning to make decisions about its future. And at this point, it's made 3 cell types and 3 cell types only. Those 3 cell types are the trophectoderms, which is the outer cells that you see here, and they are the cells that are going to go on and form the placenta. It makes a layer of cells on the surface of what we call the inner cell mass, called primitive endoderm, and they make another extra embryonic cell type. And then there's a little group of cells that are stained pink here because they're stained with a transcription factor called OCT4. And these are the pluripotent cells of the embryo. And my early work and my continuing work today is really focused on 3 major questions around those 3 cell types in the embryo. What do they give rise to? So when we see this embryo, what are these cell types actually going to produce later? And I've given you a clue already. And when is fate restricted? When do cells know that they can't go back and do something else? So when has the DNA program become restricted and restricted those cell types? How are they established? What are the clues? What are the signals that lead to the formation of these cell types? And relevant to what we're going to talk about today, can we derive stem cell lines from this early stage that we can grow indefinitely in culture and replicate the lineages of the embryo? This embryo is about to implant in the uterus. Once it implants, everything moves forward. We start to see the differentiation of cells, we start to see the formation of the body axis. We can't freeze it. But if we could capture and freeze those cells at that point in time, we would have stem cell lines that could be very useful in the future. So how did we start? So when I was a graduate student, I worked with Richard Gardner in the UK, and Richard was one of the first people to develop what we call mouse chimeras. And that is, he was able to take cells from one embryo, genetically marked, and inject them into the blastocyst of the other embryo. You see that going on in the middle here. There's the blastocyst. Remember, it's the size of a speck of dust, so this isn't easy. This is all done under the microscope with micromanipulators that you see on the bottom there. But those cells then, when you put them into the blastocyst, they can incorporate back into the embryo. And if they're genetically marked, you can follow their fate. And what Richard showed, and I showed in a series of other experiments, was that if you take the cells of those pluripotent cells, those epiblast cells in the embryo, and put them back into a blastocyst, then if they're marked here with a coat color marker, you can produce a normal mouse. But the cells from one embryo have integrated and have actually contributed to all the different cell types of the mouse. So it's those kind of experiments that essentially told us that by the time we get to the blastocyst stage, we essentially have 3 restricted cell types. They give rise to specific lineages later in development. The most important one, really, is the trophoblast, because it makes a placenta. And as a mammalian embryo, if you're a mouse or a human, you would not be here today without the placenta, because that's how you interact with the maternal environment. So the placenta is the first cell type to form. The primitive endoderm makes it the yolk sac, which is also needed for the embryo to survive in the uterus. And then it's those epiblast cells that make the entire fetus. So they are pluripotent. They can make a whole mouse. And that we knew then from mouse embryology, experimental embryology, way back from work I did as a graduate student and a postdoc. So pluripotent, that's an interesting term. What do we mean by that? We're going to go on and talk about pluripotent stem cells. Why don't we call them totipotent? Why, if I say they make every cell type of the fetus, why don't we call them totipotent? And the reason, of course, is that from these early experiments, we knew that they don't make the cells of the placenta, and they don't make the cells of the yolk sac. So epiblast cells are pluripotent, but they don't make the extraembryonic cell types. The embryo has made some restricted lineage decisions already. And over many, many years, and we're really compressing research over many years, we've gone on, and others have gone on, to show that from that blastocyst in the mouse, you can, in fact, derive 3 lineage-specific stem cell lines that remember where they came from. The most important ones are embryonic stem cells, shown in the middle. We did not derive those. We certainly tried, but we were not successful. The people who were successful were Gail Martin and Martin Evans way back in the 1980s. And they were able to show that you could capture that pluripotent capacity of the epiblast cells and, in the right culture conditions, grow cells indefinitely in that undifferentiated state. They express pluripotency markers, OCT4, NANOG, other genes that are required for specifying pluripotency. And importantly, although they can remain undifferentiated, if you put them back into the blastocyst, the same experiment that I did as a graduate student, but now with stem cells, they behave like the epiblast cells. They contribute to all cell types of the fetus, but they don't make the placenta and they don't make the yolk sac. And you see that in that image where the stem cells are marked with beta-galactosidase, so the cells are blue. The yolk sac has no blue cells and the placenta has no blue cells. Afterwards, after the embryonic stem cells are derived, my lab went on to show you could derive trophoblast stem cells and what we call Zen cells from the primitive endoderm. They can grow in different culture conditions and also remember their cell fate. When we put those back in the blastocyst, the TS cells, the trophoblast cells, go back and make the placenta, but not the embryo. And the Zen cells make yolk sac, but not placenta or embryo. So we've got a nice system that we and others have continued to study to understand the genetic and epigenetic processes that help establish and maintain lineage specificity. So that's the research that my lab and others are still involved in, and we believe that studying the embryo can help us also understand stem cells and how to do better in making pluripotent stem cells. But when we were looking and people were working on embryonic stem cells, we also asked ourselves, well, do we really know that an embryonic stem cell that's grown in a petri dish is truly behaving like the epiblast cell in the embryo? Are they truly pluripotent? Well, I told you that epiblast cells can actually produce the entire fetus. That's what they're designed to do. Can ES cells do that? Are they truly pluripotent? Can ES cells make a whole mouse? So this was a wonderful collaboration with my colleague Andras Nagy in Toronto, and in the early 90s we did a series of experiments in which we used a bit of genetic trickery to see if we could generate a situation in which the embryonic stem cells were forced to take over the entire mouse. And we did this by taking embryonic stem cells, which I told you are pluripotent but don't make trophoblast, and combined them with tetraploid embryos, which for reasons that are still not fully understood, are pretty good at making trophoblasts and extra embryonic cell types, but don't make the embryo itself. So what happens if you put the two together in an aggregation, as shown here? Those cells will incorporate and make one blastocyst. But if it's going to go on, the only way this can survive is if the embryonic stem cells take over the embryonic compartment. And that's what we found. Early on, we showed that if we had blue embryonic stem cells, combine them with green tetraploid, you could get fetuses like the ones shown here that are entirely blue. That's entirely ES-derived, supported by the tetraploid cells that are green. So as we went on and we got better embryonic stem cells, more stable embryonic stem cells, we were able to go on and show that you could actually not just make a fetus, but you could make an entire mouse. It's perfectly viable, can grow up, can breed from stem cells that have been maintained for many generations in the petri dish. And here's one litter of ES cell-derived mice. They're carrying a GFP marker, so they're all glowing green. They're little babies, they're running around, and they grow up to be perfectly normal. That was a very important experiment because it truly demonstrated that a stem cell line could retain full pluripotent capacity in the petri dish. And if you think about it, I still think about it, it's pretty amazing. So the question then became, if mouse embryonic stem cells can make a whole mouse, what if we could make human embryonic stem cells? Not to make a whole human, but to retain that pluripotent capacity that would provide an endless resource of cells that you could think about using for regenerative medicine. Because they can make every cell type in the body, you have a pool of cells that you might be able to use to derive cell types to treat many different kinds of degenerative diseases. So, can you make human embryonic stem cells? Well, the first human embryonic stem cells were derived by Jamie Thompson in Wisconsin in 1998. So the mouse cells, it was 1981 when the mouse cells were first isolated. It took quite a long time, following on from that, for people to be able to work with human embryos and be able to derive similar kinds of cells from human embryos. Similar kind of approach, taking the blastocyst, taking out those pluripotent cells from the human blastocyst and growing them in appropriate culture conditions. Jamie was able to show that, again, he could make pluripotent stem cell lines. And when I say pluripotent, how do we know, how do we prove that a human embryonic stem cell is pluripotent? Well, clearly it's not appropriate or ethically advised to make a human chimera, and we'll come back to that a little bit later on. But what people, Jamie and others, and many over many years have gone on to show is that in fact those embryonic stem cells in the right culture conditions can be driven to differentiate into pretty well every cell type you can think of in the body. And every day people are developing new ways of driving pluripotent stem cells to differentiate into cells that are useful for studying human biology, studying disease, but importantly, cell types that can potentially be used to treat degenerative diseases. So there's no question that the discovery of human embryonic stem cells opened up new vistas of regenerative medicine. But new vistas for regenerative medicine, but a big ethical debate. So when human embryonic stem cells were first isolated, there was really an uproar in the public domain, particularly in the United States, but also flowing over the border here into Canada. So why? Why big ethical concerns? Because fairly obvious. The obvious answer to this was that human embryonic stem cell work required taking human embryos and essentially destroying them, at least destroying their capacity to put them back into a woman to give birth, to generate the cell lines. So it required the use of human embryos. And for many people, the use of human embryos in research generally was not acceptable. And certainly this concept of specifically taking embryos and using them to make cell lines was anathema to many different religious and political groups around the world. So, in fact, where do human embryos come from for embryonic stem cell research? The source of human embryos for almost all the stem cell lines that have been made over the succeeding years is from in vitro fertilization clinics. IVF itself, when I was a graduate student with Richard Gardner, I was in the reproductive biology lab at Cambridge where Bob Edwards was working on in vitro fertilization. And if you remember, those of you who were around then, the first test tube baby, Louise Brown, when she was born, it was a huge uproar. We're making test tube babies. This is the beginning of the end. We're going to change how we approach human reproduction. It was very controversial. And yet, of course, IVF today is an accepted part of medical practice. 1% of all babies in North America are born after some form of reproductive technology. So IVF occurs. It occurs in clinics, and the process of IVF, where you take ovulated eggs and sperm and put them together, usually generates more embryos than can be used for reproductive purposes. Those embryos are frozen and eventually will be discarded if not used, and can be donated for research. It requires full informed consent from the donors, and it falls under legislative and other regulatory restrictions in many different jurisdictions around the world. So, very controversial. It's meant that in many jurisdictions, particularly in the US, there became then a sort of hype around embryonic stem cells, that we were going to cure disease tomorrow. That's a dangerous thing. If you tell the public that you promise too much, then that can put the whole research under some scrutiny. But in fact, it was extremely important to be able to move forward in this research. So what happens in Canada? And that's what I want to focus on today. So in Canada, as all this was going on, so 1998, the beginnings of embryonic stem cells, the production of embryonic stem cells over the next few years, was the same time that Canada was developing an act, a government act, called the Assisted Human Reproduction Act, which was passed in 2004. This was really a law designed to regulate all forms of reproductive technology. It has suffered mightily over the succeeding years, and many of the regulations that were intended to come into play to regulate surrogacy, IVF clinics, sperm donation, many of these regulations were never brought into action because the law was challenged by Quebec to the Supreme Court because it was a federal law that they felt was impinging on the provincial rights to regulate healthcare. So Quebec challenged it, which meant that all the regulatory capacity around the actual human reproductive technologies has fallen back into provincial hands. But remaining in this act, which stands on the statutes today, are some criminal prohibitions. So many of the criminal prohibitions that remain in force in the Act are related to research, because research is a federal responsibility. And so the criminal sanctions that exist actually deal with embryo research and embryonic stem cell research. So for embryonic stem cells, and I was involved in many others and a lot of discussions at the time, this Act, we have come through the Act with a situation in which human embryonic stem cell research is possible in Canada. It does allow research on existing embryonic stem cell lines, and it does permit potentially the derivation of new embryonic stem cell lines from IVF embryos, as I just described. On top of the Act, and importantly, the Canadian Institutes of Health Research has set up a set of guidelines. There was a task force set up by AHRA at the time, which I chaired, trying to get ahead of the game, knowing that there were going to be criminal prohibitions put in place, we wanted to have a regulatory environment set in stone through CIHR so that we could convince the government and indeed the public that Canadian scientists were going to be able to do this research, but in a clear, open, transparent, and regulatory regime. And so the Canadian Institute of Health Research guidelines still exist, and they've been taken into the Tri-Council policy statement. And that oversight committee is now set up. There's now an oversight committee at CIHR for which, if you're doing any work with human embryonic stem cells, you have to go through what's called SCOC and get permission to go forward. So there is a national oversight committee for embryonic stem cell research in Canada. So that's fine. We have the guidelines for general embryonic stem cell research. But what's left in that act? And what are today's implications? So I'm going to go through some of the criminal prohibitions. And I'm smiling because I'm reminding you that these are criminal prohibitions. So that means that if you were to do this, you could be thrown in jail or have a very large fine. There have been nobody's been taken to court or arrested under this act as far as I know, but it still stands and it's important. So what is banned and what are today's implications? So let's just go through. Here's one. Number one. These are the bans. Number one, create a human clone using any technique or transplant a human clone into a human being or into any non-human life form or artificial device. So create a human clone. What does that mean and why was that sort of rolled up into the act? Well, it got sort of worked up into embryonic stem cell research at about this time. If you remember, cloning Dolly the sheep, there she is at the top. Cloning is a technology first developed in frogs by John Gurdon, who was my mentor when I was an undergraduate at Oxford. And John Gurdon showed that if you took the nucleus from an adult cell that had all the DNA information and put it back into an enucleated egg, that DNA information could be read out and you could reconstitute the whole of development. That was very important because it said that it's possible to reprogram the adult genome to be able to start development all over again. And that applied to mammals led to the formation, the first birth of a mammalian clone, which was Dolly the sheep. And that's reproductive cloning. So there's a lot of concern worldwide about the slippery slope. If you can clone Dolly the sheep, what would happen if we started cloning human beings and made a race of identical people, etc.? So worldwide, there's pretty well a concerted ban on reproductive cloning. However, there has been some interest and importance to use the cloning technology to generate stem cells. And that's what's shown on the bottom here is has been called therapeutic cloning. We would call nuclear transfer stem cell production. And that's the idea being that you could generate patient-specific stem cell lines by taking a nucleus from a somatic cell of a human, putting it into an enucleated oocyte, allowing it to get to the blastocyst, but only the blastocyst stage, and then instead of doing reproduction with it, putting it back into the womb, you would take that blastocyst and make embryonic stem cells. The idea here being that you potentially could be making stem cells that would match a patient who might need them for regenerative medicine, but also you could take it from patients and make stem cells that would mimic specific diseases. So this was at the time an important approach. There was no other way to think about making patient-specific stem cells in 2004. And yet in Canada, it would be completely banned. This ban would not allow you to create a human clone by any technique. That doesn't mean just by reproduction. It actually, and the Health Canada continues to interpret this way, that this would stop you doing the technology at all, even to generate a blastocyst. So it's banned in Canada. So is it possible? Has it been done elsewhere in the world? So nobody, to our knowledge, has made a real human live clone, but it has been possible to make human embryonic stem cells using nuclear transfer embryo. And there are some pros and cons to doing that, and there's some reasons why people think still that this is an important technology. It's not been widely used, and the reason for that is going to come up in the next slide, because it's been overtaken by other approaches. But it's also true that the technology of doing that nuclear transfer, being able to work with the eggs and transfer nuclei, is actually extremely important as a technology, which I'll talk about again in a minute, to treat mitochondrial disease. So the ban in Canada is putting a block on a number of different technologies going forward. So why have Canadian scientists not been up in arms about not doing nuclear transfer? Well, because the game changed. I'm sure many of you are aware of this game changer. This was the discovery of induced pluripotent stem cells by Shinya Yamanaka in Japan, 2006. So we're now 2 years after the Reproductive Technologies Act. No embryos are used in this approach because what Yamanaka showed was that you could take an adult cell, whether it's a skin cell, a blood cell, even cells actually from the urine. There's enough cells floating around in urine, so easily accessible cell types. And by adding back just 4 genes that we know are critical for pluripotency in the embryo and in embryonic stem cells, he was able to turn back the clock and turn those skin cells back into what are called induced pluripotent stem cells that behave just like embryonic stem cells. They can differentiate in the culture dish into all the different cell types, but importantly now, they open up new possibilities because they, like the nuclear transfer approach, but in a much easier manner, allow us to think about modeling human disease in the petri dish. We can make stem cells from anyone here in the audience. So just stand up and I'll take your cells. And we can do it from patients with different genetic diseases and begin to mimic and study the disease and use those cells to actually develop new drug screens and new therapeutics. So extremely important tool. In the long run, and I think this is a little further off, this is going to be an important approach, again, for rejection-free transplantation, because these cells could potentially be made to match the patient who needs them for regenerative medicine. So here are the two of my heroes of science, John Gurdon, who, as I say, was the first person to clone a frog and showing you could reprogram the adult nucleus, and then Shinya Yamanaka, who many years later then was able to show that you could directly reprogram an adult cell. And they shared the Nobel Prize for Medicine in 2012. But just a little plug for the Gardner Award. The Gardner Award always wants to be there first, so Yamanaka received the Gardner Award in Toronto in 2009, and here's my husband and myself with Shinya and his beautiful wife receiving the award in Toronto in 2009. A true game changer, and in a very short period of time, you see the first paper was 2006, the Gardner Award in 2009, and the Nobel Prize in 2012. So where are we at with pluripotent stem cells today? So research is going on. iPS cells open up new vistas and certainly are not affected in terms of regenerative medicine by the Canadian Act. So work is going on with both cell types in Canada and around the world. So all that hype around pluripotent stem cells in the early days because of the controversy, has it really paid off? And the answer is we're moving. There is movement towards the clinic with cells derived from pluripotent cells. It's going to take time. There are safety issues. You do not want to rush into patients with these cells that have the potential perhaps to make tumors. You have to be careful. You need to be able to make stable cell types. You need to understand how to drive them into the appropriate cell types. And you need appropriate clinical protocols to be able to put the cells back into patients. But here are some of the ones, my favorites, that I think are moving forward and we will see come into clinic soon. Spinal cord injury. There were some trials from Geron early on. They've restarted using cells derived from embryonic stem cells, not to replace the nerve cells that are damaged in the spinal cord, but to remyelinate the existing cells and try to alleviate damage. So this will be an adjuvant treatment, but important. Macular degeneration, a rather specialized but important one, where the retinal pigment epithelium degenerates. And it turns out that you can make RPE very easily from embryonic stem cells, and there are trials, several trials going on with both embryonic stem cells and iPS cells, derived cells, underway right now. Small trials, we'll see what happens. Parkinson's, an important target, a very important disease. There are production of dopamine neurons from embryonic stem cells is now really well underway, and there's an international consortium that is moving rapidly to try to move this into phase 1 trials. Diabetes, producing insulin-producing cells, not a whole pancreas, just the cells, the beta cells that produce insulin in a glucose-dependent manner. Good advances worldwide in producing those cells. And importantly, here in Canada, in Edmonton, we have fantastic groups that have shown already the clinical protocol to be able to take islets into patients to treat for type 1 diabetes. And those same groups are now working with companies that are developing progenitors and testing them in clinical trials. Heart disease, lots of trials with different kinds of stem cells for cardiovascular disease, but none of them are really replacing the damaged cardiomyocytes. Exciting data, preclinical data with cardiomyocytes from embryonic stem cells into preclinical models, suggesting that it may be possible to actually repair hearts. And a lot of that work is going on in a number of places, but a very big and very big group here in Canada, in Toronto, which has just actually attracted a very large industry investment. So, moving forward. So let's go back to the ACT, though. What else has it got? So embryonic stem cells, we're okay. With regenerative medicine is okay. But there are still some other things that are very important, are right at the frontier of where science and clinical application is going. What else does the ACT say? Number F is, no person shall knowingly alter the genome of a cell or a human being or an in vitro embryo such that the alteration is capable of being transmitted to descendants. That means no germline gene modification. You cannot make a genetic alteration in a cell such that the offspring of that person would carry that alteration forever. This is an important one, because this is where science is changing. First of all, this one applies not just to the nuclear genome, but also to mitochondrial genomes. So the mitochondria have DNA, so when we grow up, our mitochondria are inherited through the mum in the egg. And there's now, using nuclear transfer technology, there's a big push to actually be able to use nuclear transfer to treat mitochondrial disease. And the general idea is shown here, that if you have a woman who has unhealthy mitochondria, carries mitochondria that are known to cause disease, then what you want to do is to take out the nucleus from the mother's egg and put it into a donor egg that has good mitochondria. So there's nothing wrong with the genome of the nucleus, it's simply the mitochondria are bad. And so the idea then is you do that, you transplant the nucleus and fertilize it with the father's sperm. Genetically, in terms of the things we most think about that are encoded by the nuclear genome, this is exactly the same as the starting situation. But now the mitochondrial genome is derived from a third parent. So this has been called three-parent babies. Again, it's much better, I think, to call it mitochondrial replacement therapy. But it does have this concept that you have three parents. And where are we at with this? This is happening worldwide. The first reported baby, and this is not published in the peer-reviewed literature as yet, but from a reputable group, however, carried out in a Mexican clinic, the first baby was reported just very recently in Mexico. The procedure, however, has been approved to treat mitochondrial disease in the UK, not yet performed, but it's been approved. In the US, the FDA is banned from even considering protocols. So there's been from the House and the Senate, there's been a ban. The FDA, if somebody comes forward with this, you're not even allowed to consider it. So it's not going to happen in the US. The National Academy had a working group looking at this, and they came down with a very interesting, but also, I think, ethically concerning recommendation, suggesting that mitochondrial replacement therapy could go ahead so long as you did it on male embryos only. Think about it. Why would you do that? Well, because the male embryo, the mitochondria are inherited through the egg. So a male embryo would not essentially carry on the mitochondria into the next generation. I think that's ethically concerning. Why would you choose male babies and not female babies? As the Act is read here currently in Canada, it will be a total ban. Not sure whether the National Academy recommendation would get us by that, but again, I think it brings its own ethical concerns. So mitochondrial disease therapy, not available in Canada. Okay, let's go back to the same codicil, altering the genome of a cell of a human being to transmit to descendants. And as I say, this was really directed towards altering the nuclear genome, and it would apply to both correcting genetic disease, what we might think is a good thing to do, or genetic enhancement, making stronger people, making blue-eyed babies, etc. So that was in the Act, and there's been some consideration about that. There's a general feeling that maybe this is a bad thing to do, but it wasn't really considered very seriously because we didn't have good technologies to even think about doing this in a practical way in human embryos. Now we do know how to do it, and we've known how to do it for a long time in the mouse again. We've been able to make transgenic mice, transgenic animals, adding DNA, adding genetic sequences, altering the genome, having it inherited in embryos by zygote injection, by taking embryonic stem cells and altering genes in them and making those stem cells into chimeras that can contribute the germline really since the 1980s. And it's extremely important technology. It's allowed us to explore the mouse genome and hence, by analogy, the human genome in extremely in-depth and important ways. But it's the mouse. And the process is not particularly efficient. Trying to translate that into what you would want to do in human embryos, where you only have a few precious embryos to work with, really was considered unlikely to be approachable and certainly not safe to go forward. So why is this back in the news? Well, it's back in the news because we have another game changer. And the game changer is new technology to edit the genome. And this is what's called CRISPR-Cas9 gene editing. This is a gene editing system that works in bacteria to protect bacteria from invading viruses. And it works by targeted cutting of the DNA and allowing direct replacement at very, very specific sites across the genome. Not specifically really different from anything we've done before, but the approach is highly specific to the nucleotide and extremely efficient. And you can use this technology, and it is being used extraordinarily widely, rapidly, to modify plants, to modify animals, to modify bacteria, to modify fungi. And it's rapidly being used and thinking about moving to the clinic for gene therapy in humans, somatic gene therapy. So you could actually treat cancer, so CAR T therapy, which you may have heard about, immunotherapy, modifying T cells so that they can respond and kill tumors. That's being done with CRISPR. Treating hematopoietic stem cells and correcting sickle cell disease, correcting beta thalassemia, putting those cells back into patients is being thought of, is moving rapidly to the clinic. HIV, protecting people from HIV by knocking out the receptor. Even Duchenne muscular dystrophy in the mouse, and remember we do have a good record at curing mice, but in the mouse it's been possible to take the CRISPR-Cas approach and carrying it in an adenovirus, get enough repair of the muscle cells in a Duchenne mouse to see improvement in muscle function. So this is amazing, and it's so efficient and so specific that it's changing the way we think about gene editing. And again, extremely fast, and the Gardners are there first. So last year, 2016, which was really the gala we just had in October, my first gala, you see me there on the left, we gave all 5 international awards for the Gardner Foundation in 2016 to what I've called here the CRISPR-Cas9 quintet, the 5 people in the middle here who between them have developed the bacterial system and translated it into human cells and eventually into human treatment. So again, extremely exciting, and we had a very exciting gala. So what about the germline? So if you can do it in somatic cells, again, nobody's going to object to that. But does this open up the possibility that we could modify the human germline? So here we have a little picture of what you might potentially do to a human baby, designer babies. Importantly, we could fix cystic fibrosis or Huntington's disease. But what about if we could, make a better sprinter? Maybe we could improve eyesight, lower risk of disease. That might be good. Could we alter IQ? Well, luckily we don't know enough about the genes yet, but one day we will. So where do we draw the line? And this has caused, again, a huge ethical debate around the world. Many different organizations and agencies have had working groups to consider where you might want to put an ethical boundary. If you think about it, it's pretty hard. The only consensus that seems to come out is that using gene editing to actually study the biology of the human embryo in the petri dish is something that would make sense. Understanding all those events that I know about in the mouse embryo, we need to know how it works in the human embryo, again, so we can look at the similarities and differences and use it to understand stem cells. So there are 2 groups, at least, approved around the world. Kathy Nyakin in the UK is using gene editing to study lineage development in the human embryo. And my ex-postdoc, actually, Frederick Lanner at the Karolinska, has also been approved to use this approach, but not to put those embryos back. This is pure research. So international groups, there was an international summit on human gene editing, which was a very important and wide-ranging discussion. There am I sitting on the stage with a couple of other leaders in this area. And at the end of that, the statement that came out reflects what's coming out from many working groups around the world as it stands today. And it's a kind of, no, we can't draw a line. We don't know, unless we say absolutely nothing, we should not do it, which is what the Act says. Given the technology, we need to think about this very carefully. So it says it would be irresponsible to proceed with clinical use until we've got safety and efficacy, for sure. That's the scientist speaking. But number 2, that there's a broad societal consensus about the appropriateness of the proposed application. And there has to be some appropriate regulatory oversight. And at present, these criteria have not been met. And I think that's true everywhere, with perhaps the exception of the UK. So we are in a situation in Canada where we have not had an open discussion, where it is currently criminally banned to even think about it, and probably not clear, Health Canada needs to rule on this, it might even be banned just to do embryo research in the petri dish, but certainly for germline editing. So this is a concern that will, I think, lead us at some point to go back and reconsider the Act. A couple of other things, just quickly. The science is always ahead of law. It's a big mistake to put things hard in law. Number B, no person should knowingly create an in vitro embryo for any purpose other than creating a human being or improving assisted reproduction. No creation of an embryo in vitro. Okay, maybe we don't want to create an embryo, but maybe we have new ways of thinking about creating an embryo that don't even require us to use eggs and sperm. So there's no issue of donation or informed consent, because there's rapid science advances in the mouse in generating eggs and sperm, not from gametes, but from pluripotent stem cells directly. So several groups now have been able to take embryonic stem cells and generate functioning spermatids. So embryonic stem cells are taken in culture, differentiated into early mesoderm, given the right signals, they make pre-germ cells, and the germ cells, given enough appropriate environment and interactions, can make spermatids that can fertilize an egg after injection. And then just very recently, in October, a Japanese group showed they could do the same thing for the female germline. Those oocytes you see at the top there were derived from embryonic stem cells in culture, and the mice at the bottom were derived from fertilizing those oocytes with sperm. So amazing. You can go from a stem cell to a functioning egg or sperm. Nobody, to my knowledge, has done the obvious experiment yet, which is to take an egg and a sperm derived from stem cells and put them together. That would be truly creating an embryo in the petri dish. And it's not yet been successful with human stem cells, but presumably this is just a matter of time. So this is going to change and require a whole lot of different ethical concerns. And then finally, what else does the Act say? No person shall knowingly maintain an embryo outside the body for more than 14 days following fertilization. So you're not allowed to culture an embryo beyond 14 days. So you can, if you make a stem cell line, it's no longer an embryo, you've destroyed its structure, but if you want to keep it intact, you can't grow for 14 days. This 14-day rule is actually applied worldwide. It's become accepted very early on in the system. And why? Number of reasons, but it marks the time in the embryo when the first body axis forms and the nervous system begins to develop. And again, it's a sort of somewhat arbitrary line. And there was no culture system available to even think about getting to an organized structure in a human embryo. So again, the restriction was there, but it didn't cause a big concern. Last year, now we're in January, 2 groups published papers in which they were able to take human embryos and grow them over that implantation period and begin to form structures that start to show organization and not the body axis, but get towards the process of gastrulation. Now, you do need a little imagination to take the pictures on the top and reproduce them into what's shown on the bottom, but with some imagination, you can tell that this embryo is starting to organize. And again, improvements will only get better. We'll be able to have better culture systems to grow them over this implantation period. So approaching the primitive streak, approaching gastrulation, is this a specific line we should not cross? And I had a commentary on this in Nature at the time. So you see the blastocyst, and on the bottom you see in the normal embryo, it organizes, makes the amnion, starts to make the primitive streak. And what they're seeing in these rather flat cultures is a structure that is beginning to look like the post-implantation human embryo. So is this something that needs to be reconsidered? Do we need to go back and look at the 14-day rule? Because these are obviously very close to being able to get through the primitive streak and give us new insights. So should we be doing that? Why would you want to do that? And this is to be discussed, but here are some reasons. First of all, to be able to study human gastrulation, there's no other way of understanding that. You cannot access it in the human itself. It's a key stage. It's when everything happens that sets in place the formation of the embryo. And there's a nice radio lab, if any of you like podcasts, there's a really nice radio lab on the primitive streak where their reporters go and visit these people and go, it's amazing, just think about it. Formation of a human primitive streak in the embryo. So just the fundamental knowledge of our cells is important. It is important because of these differences between mouse and human. It's a time when the germ cells form, so this would help understand germ cells and infertility. And it would allow you to really study implantation, which is really one of the key times of early embryo loss. So understand placental formation better. And using CRISPR approaches, we could experimentally begin to perhaps understand the genetics of some of these early developmental defects. So issues to be discussed. Finally, I forgot one. Chimeras, back to the beginning, I made chimeras. We've made mouse chimeras. The Act says you can't create a chimera and translate it into a human being. And it really is about human-human chimeras. And I think everybody is okay with that. We're not going to make human chimeras. But the Act is not clear about human-animal chimeras, but the AHRA guidelines and the Tri-Council statement are very clear. It says you cannot take human embryonic stem cells, germ cells, induced pluripotent stem cells, or other cells that are likely to be pluripotent and combine them with a non-human embryo. You can't even combine them, and you certainly can't put it back and have it complete gestation. So why is this a concern? Well, there's research going on worldwide on this as well. These interspecies chimeras have been around a long time. I made one of the first ones years ago between 2 species of mouse. So it's possible to cross the species boundary and get normal development. More recently, Hiro Nakayuchi in Japan and now in the US has been making rat mouse chimeras and developing a proof of principle that you could generate whole organs in an interspecies situation, because what he did was make a mouse that couldn't make a pancreas and put rat embryonic stem cells into that blastocyst. And then what happens is the stem cells distribute all over the embryo, but it's a bit like that tetraploid complementation. The pancreas can only form if all of it comes from the rat cells. And he was able to do that, generate then a pancreas that's derived from the rat. Think about that translated into human stem cells. And this is his vision, that the rat is probably not a good model. Let's take a preclinical model that's similar in its physiology, size, etc., to the human, and think about taking a pig and making it apancreatic, putting in human iPS cells and having them generate a human pancreas in the pig. So the idea is not just to take stem cells to generate cell types, but to generate real organs. This is moving forward. There's going to be another paper published this week on this, so watch the space. You're going to see it in the newspapers, I'm sure. And so this opens up new ethical issues, for sure, but it does open up new concepts and new possibilities in stem cell research. So here we are today. The science of stem cells is amazing. It's moving apace, but the ethical, legal, and policy implications are way behind, especially in Canada. And I do think that we're going to see a need to have a broader discussion as we move forward. So it begins with blastocyst, and we continue to explore today. Thank you.
Thank you very much, Dr. Rossant, for a fascinating talk on the age of science and ethical dilemmas. We have the time for a few questions. So maybe I ask you,
Sure.
So where do you, I mean, you basically in your last slide, you said that the regulation lags behind, especially here, and that of course implies that we will lose the contact with what's going on worldwide. How do you think that can be changed?
Well, I think it's a good question. I think Health Canada is the first place that we have to look to. They are very interested in looking at the regulatory domain here, and certainly we've had ongoing discussions with them. It's very hard to reopen criminal law. Nobody wants to go back and revisit the law, because that means going back to Parliament, and it's very difficult to do so. So I think it's going to take some time, but we will have to work together. It's not in all areas. You can see that some of them, maybe we don't need to be cloning, that's okay. But do we think about mitochondrial disease? Yes. Do we think about potentially these chimeric approaches? And then there are people who would like to do that kind of work. Growing the embryos in culture, I think these are things that need to be discussed. So there's a need, and I think a number of different groups are going to have to come together to do that. But it's not going to be easy to change what's in the law.
Okay. There's a question over there.
Yeah.
In the U.S., there seems to be patient groups who are very interested in mobilizing the utilization of CRISPR. I haven't seen that in Canada, but I just wonder, whose obligation do you think it is to try to bring this to the forefront and make sure that it's addressed?
Well, so yes, you're right. Patient groups, not just for CRISPR, I mean, patient groups are also very active in the sort of stem cell arena as well to try to get novel treatments through to therapy. So the CRISPR, of course, approach for somatic gene therapy can occur here and will occur here. The germline at the moment is off the charts. But yes, I think part of these discussions, and there'll have to be some sort of broader consensus conferences that would include patient groups as well. The global forum that was held in Washington is a good model, because that actually brought together patient groups, scientists, policymakers across a broad range to really discuss the issue. But at the end of the day, even that one came out with a good consensus statement. But in the US, although there is no law banning all of this, there's very strong regulatory pressure on the FDA, who would have to approve these. And again, the power of the, I don't know what you want to call it, but the extreme right is really preventing any of these technologies being even considered by the FDA. So there's a strong lobby against this, but also a strong patient group trying to battle that. So that because everything is more polarized in the States, I think you see it more active there. People somehow think it's all right in Canada. It's not all right in Canada.
Last question.
Do you think you're going to be able to keep on regulating this? Don't you think at a certain point there's going to be like hacker science that are going to be doing it somewhere and it's going to happen, and it might be better to just do it in a controlled way?
Yeah, so can hackers get into this? So people talk about CRISPR being something that you could do in your garage. It's not that easy, and germline editing would not be that easy. Do I think that it will occur worldwide? Will people do germline editing in human embryos? Yes, I do. Do I think that means we should just throw up our hands and say, well, it's okay if it happens? I don't think it is. I think that it's much better to develop a regulatory environment to to set up a system whereby any application that people want to use gene editing is brought before a regulatory body that considers the pros and the cons. And that's what happens in the UK. So mitochondrial therapy, for example, has gone through a whole layers of discussion through the HFEA before they really considered that the science was ready, that the ethics were correct, and that it was really clinically applicable. And again, remember, that's only a small group of patients, and still it took a long time to come up with that. And I think that's going to be true for every application of gene editing. Each one is going to have to be considered on its own merits. Okay.
Thank you very much again for a fantastic, stimulating lecture, Dr. Rossant.